Okay, hey guys. We are on chapter 10, which is on DNA and making proteins. So just a quick reminder, we've talked about it before, but again, I wanted you to remember that proteins make up everything in our body. Everything from hair and skin color to if you're lactose intolerant, how your organs function. Everything in our bodies pretty much comes from making proteins. And the instructions for making those proteins comes from the DNA, okay? So just like building a house, we have to have our blueprint, which is the DNA, and then that blueprint gets turned into the actual finished product. So what we're going over today is how that process works, how we go from DNA to protein. There's some vocabulary. I'm actually going to just write this one so I can draw in between. Again, this is chapter 10. And let's go ahead and look at some vocabulary. The first word is transcription. This word obviously has definitions outside of the science world, but it's a science definition, that I'm worried about, or that we're going to go over today. So transcription is basically the process of converting information from DNA to RNA, okay? Then we have the second process that's called translation. And again, obviously there's countless different ways to use this word. In biology, translation is going from the RNA information to the actual protein. So what we have here is this kind of sequence going from DNA to RNA to protein. I'm going to add to trait. And again, the trait is the physical representation of the protein. So the process of DNA to RNA, this is transcription. And the process of RNA to protein is called translation. Okay? All right, so why do we do this? Why do we go from DNA to this intermediate RNA? Well, I want you to think about the DNA as being a master copy of something, okay? We don't want it damaged. We don't want it destroyed. We don't want to increase its risk of having something happen to it. So the DNA stays really protected in the nucleus of the cell. However, to build proteins, the genetic information has to leave the nucleus and get into the cytoplasm of the cell. And again, we don't want the DNA to do that. We want it to stay tucked away in the nuclear vault, if you will. So instead, we're going to have this intermediate form come in, and it's going to make a copy. It's going to make a copy of the DNA, and then that copy is going to be what goes out into the nucleus, or I'm sorry, out of the nucleus and into the cytoplasm. The copy is RNA, okay? That copy is then going to be used to build proteins. So let's say, go back to that building a house example, and there's one person that has the original master copy of the blueprints, but those get kept under lock and key. We don't want anything to happen to them. But we've made copies. We've made copies of the master that get handed out so that the house can be built, okay? Those copies, again, are the RNA, the master being the DNA. The house is the finished protein, or the trait, okay? All right, so a couple of differences between RNA and DNA. I'm going to switch over to my other screen here. So DNA is a double-stranded molecule. So it looks like a ladder that you've twisted. Okay, it's called a double helix. The backbone, the big blue dots, the little red dots are sugar and phosphate. And then the bars or the rungs of the ladder, it looks like they're using orange and blue and yellow and green. Those are the nucleotides that build those are where the genetic information is actually stored, okay? So if I'm gonna look at RNA then, here's the difference between DNA and RNA. Just a second, get this to be a little bit bigger. Okay, so here's RNA. It's single-stranded. It's not the double-sided ladder like we have for DNA. And it has nucleotides as well, those rungs of the ladder. But notice that there's only one side on this one, right? It still has a sugar and phosphate backbone, just like DNA does. The sugar is just different. But again, there's only one nucleotide rather than having it attached to another one. And this is because RNA is the copy, right? DNA is the master, but RNA is the copy. And so it doesn't need to be both sides of the DNA. We actually only needed to make a copy of one side of that DNA, okay? We'll get into that a little bit more in a second. You can kind of see the difference between DNA and RNA here. This is actually showing the transcription and translation process going from double-stranded DNA to making a copy of just a single strand RNA. And then from there, we're going to use that RNA to build the proteins. Okay, we're going to build the house off of it. Okay. Let me go ahead and show you, actually I'm going to write down here, some of the differences between DNA and RNA that you'll need to know. Actually, I'm going to just kind of do this as a little chart, I think. Okay, so DNA is double-stranded. Right, the double helix. RNA is single-stranded. The sugar in DNA is called deoxyribose. And in RNA, it's just ribose. That's where these molecules get their names. DNA is deoxyribonucleic acid. Deoxyribonucleic acid. Whereas RNA is ribonucleic acid. Okay? DNA has 4 bases: adenine, thymine, guanine, and cytosine. They get paired together. They have a buddy system. So adenine and thymine are buddies. They're always together. And guanine and cytosine are buddies. They're always together. If you have a hard time remembering which is which in DNA, remember AT. Adenine and thymine go together. RNA also has 4 bases. Notice that 3 of them are the same as they are in DNA. I have adenine, guanine, and cytosine that are the same as DNA. The one that's different is uracil. So in RNA, there's uracil instead of thymine. But the pairing is the same. This time A and U go together, but G and C still go together. Okay? So here's some of your main differences between DNA and RNA. There's 3 types of RNA that we're going to talk about. I'm just going to add them over here, kind of to this side. And then I'll go into what each of those types of RNA do in a minute. But we're going to be looking at mRNA, rRNA, and tRNA. Okay. All right, so there's some information about DNA and RNA. Now let's actually talk about the process of going from DNA to RNA. So I have my DNA here and my nucleotide bases. Those nucleotide bases are ATGC and DNA, adenine, thymine, guanine, and cytosine. If you remember all the way back to chapter 3, nucleotides are the monomers of nucleic acids, so they're the small building blocks. So here's my double helix, although I don't have it wrapped up, I don't have it twisted, right? It just looks like a ladder. The first thing I'm going to do for us to go over, I'm just going to give a random sequence here. Okay. Now, since in DNA we have these double-stranded ladder, if you will, we need to match the base pairs because it's a buddy system in the DNA. So I've got an A here, which means what goes with A? Which nucleotide? At, right? So this is going to be T. What goes with guanine? Cytosine. Cytosine, guanine, thymine, adenine. Okay, I'm just going to build this out here. Okay, that's how these complementary base pairs work. That's what they're called. Okay, meaning our AT and our G to C. Okay. All right. Well, when it's time to make a copy of a gene, our DNA has to unwind. Normally, our nucleotides in DNA are held together by little hydrogen bonds. That's what my purple line will be. These bonds have to be broken because we've got to get into this DNA and make a copy, but we only need one side of it to make the copy. Let's say that our body is replicating skin cells, which it does all the time. And we need to get into the genetic code to get the blueprint to make more skin cells. Well, when we're going to do that, the body is only going to take that gene for the skin cells and replicate it. It's not going to replicate our entire genome. It's not going to make an exact copy of the entire DNA strand. That's a waste of time, energy, and resources, and our body's super efficient, and it's not going to do that. It's only going to make a copy of that gene specifically. Okay, so it's got to get into the DNA, and it's got to unzip the part of the DNA where the skin cell gene would be. So let's say that I'm looking at that right here. Okay, this is the skin cell part. Well, I got to go through, and we have an enzyme that breaks apart those bonds and unzips the DNA because we want access to this strand. This strand is called the template strand. The template strand is the one we're going to make a copy of, okay? Because remember that our DNA is in the nucleus where it's very well protected. We're not going to let it go out to the cytoplasm to have the protein made off of it. We need to make this copy. And the copy if you remember from what we were just going over, is RNA. Okay? All right, so I'm going to, actually I don't want to clear them all. Clear some of these. Okay. So let's go back here to my original strand. We've unzipped it, and I'm going to make a copy of the template strand, which is the one I wrote in black letters. Here comes my RNA. Okay, and this is, oh, I can't draw a straight line on this thing to save my life. Probably because it's not straight, and I keep trying to adjust. It worked, but I had to stick my tongue out first. I'm such a dork. Okay, how many do I have? 1, 2, 3, 4, 5, 6, 7. Okay. So this is my RNA, and we're going to come in here, and my RNA is going to make a copy. I give up. I give up with this thing. I just tried to erase the bottom of it, the whole thing. Just give me a second. This is apparently like harder than anything else I've done today. There. Okay. So my RNA. My RNA is going to make a copy of the template strand by matching the base pairs. Okay. Remember that in RNA, the T has been replaced by U. Instead of thymine, we have uracil. We just need to keep that in mind as we're filling out the base pairs. So for instance, if on my template strand, it's an A on DNA, I know that its partner is going to be a U in RNA. But then I've got a G, and that's going to be a G, and this is still going to be an A, but these two are going to be U's. Okay? All of a sudden, I have my template strand. Or I'm sorry, I have my copy of my template strand. What I just did here, this process, this was transcription. Okay, that was transcription. Use the template strand on the DNA to make ourselves a copy of RNA. Now, the DNA is going to get zipped back up again, so it's going to close up. It's going to stay there in the nucleus, but the RNA is going to leave, and it's going to go out to the cytosol and to ribosomes for the translation process. Okay? Remember how I told you that there's 3 different kinds of RNA that we're going to talk about? Well, here's the first one. This specific type of RNA, the one that made the copy, is called mRNA, or messenger RNA. That's what the M stands for. Okay. It's taking the DNA message out to the cytoplasm so that proteins can be built. Okay. All right. So here it is again. I know I keep repeating it, but it's a good way to remember. Transcription, DNA strand. DNA strand gets unzipped so that we can access the gene that we want to make a copy of. Once the DNA strand is unzipped, the mRNA makes a copy of the template strand, okay, by matching the base pairs. The DNA zips back up, The RNA now is the copy, and it's going to leave the nucleus and go out into the cytoplasm to a ribosome, where the actual protein assembly is going to take place. That's transcription. All right, well, fantastic. Let's move on to translation, shall we? Let's see here. Let me take out some of this. All right. So now we're going to meet the other types of RNA. I'm actually going to add another base pair down here. So I'm going to give you another term. Let's look at the term codon. Okay, actually, let me say this differently. I'm going to call it a sequence. Sequence of 3 nucleotides. So this is a codon, this is a codon, and this is a codon. Okay, all of these are codons. So what's the point of these? Well, this is how RNA gets read. When it's time to build proteins, the way that the cell and the ribosome are able to build the proteins is by reading the codons, because these codons stand for amino acids. Okay? If you remember, amino acids are the monomers of proteins. They're the building blocks of proteins. So when we have a bunch of amino acids that we string together, we get that polypeptide chain, and then as all of those start to fold in on each other, we get the finished protein. Okay? So each of these codons code for a specific amino acid. Okay? All right, so I'm going to take this and I'm going to flip it onto its back here so that I can. And now I'm going to write the codons underneath here. What did I do? UC2. Okay. All right. So just introduced some more vocabulary that we need to deal with our translation process. All right. Now this is still my mRNA strand down here. Okay. And it's gone out into the nucleus, out of the nucleus into the cytoplasm. And it's going to find ribosomes. Ribosomes are actually made out of 2 kind of subunits. Okay, the big subunit and the small one. And our mRNA gets read by going in between those subunits. Okay? As the protein reads this, it's going to bring in another type of RNA called tRNA. tRNA stands for transfer RNA. And that's because it transfers amino acids based on the codons in mRNA. Okay? All right, the third type is rRNA, which is ribosomal RNA. In other words, it makes up ribosomes. Okay, so we've got our mRNA down here that goes through the ribosomes, which are made of rRNA, and then have amino acids built based on the codon from mRNA, with having tRNA come in and attach those amino acids. Okay. All right, let me show you real quick an amino acid chart. Okay. Here's our amino acid chart. Let me see if I can write down. Did I close my whiteboard? Shoot, I don't know what my sequence was that I did. It's all right, I'll just make one up. Okay. So let's say that my mRNA sequence, I'm actually just going to draw it over here. Okay. What did I have? It was like something like that, right? Remember that we're looking at codons. So we're looking at these three, and then these three, and then these three. Let's go find which amino acids go with these codons. So my first is UGC. Okay, so what we're going to do is we're going to start by reading across the top. We find our U column, which is, actually we're going to do the side first. We find our U column, which is here. Okay, so notice that the first letter all the way across is U. Okay, then we want to find our second position, which in this case is G. So I've got U, G. All right, here's the UG square over here. And then I need UGC. This is the third position. There it is, UGC. This codes for an amino acid called cysteine, which we're going to abbreviate right there. Now my next one is AAU. Here's my A. Here's my second A. AAU. And then my last one is GGU, which is over here, glycine. Okay? What I've just done is used my mRNA strand to have my tRNA in the purple bring in these amino acids that match what's on my mRNA. And then the rRNA, the ribosome, is going to hook those amino acids together. Okay, so this is my ribosome, right? I'm going to draw them kind of separate. And I have my mRNA going through this, and I have my tRNA dropping in my amino acids. The rRNA is going to create covalent bonds between all of these different amino acids that get dropped. Those are those peptide bonds from chapter 3. So as we string amino acids together, We eventually get a long chain, which is our polypeptide. And then from there, we get our protein. Okay. All right. Yeah, I did a totally different sequence. But that process we just did from our mRNA, it's beautiful mRNA, isn't it? From our mRNA to that sequence of amino acids that I drew on the amino acid chart, that process, this was the translation. Each one of these are amino acid. Okay. From here, it's pretty simple. Again, you link a bunch of amino acids together and they form a protein. What happens with that protein next? Well, it's going to create the trait. Okay. So basically the sequence, again, I know I wrote this at the beginning, but DNA to RNA, right, to protein to trait. And again, this is transcription. And this is translation. Okay. All right, I'm going to stop this video here, and then we're going to talk about mutations in the genetic code.